TOT
is on.
and potential public health impact. So also the YouTube version is on now. Ade, did you manage to be online? We don't see you online, so we better start the video with the presentation, and I hope that Ade manages to get online during the session. There was a problem with electricity supply in Addis Ababa.
Good afternoon. I'm Ade Desta from Addis Ababa University in Ethiopia. This afternoon, I will be talking about agro-industrial effluents in peri-urban Ethiopia with respect to microbial ecology and potential public health impact. Agro-processing is considered as an important means for inclusive economic transformation in Ethiopia. The agro-processing sector includes processing of food, drinks, leather, textile, and others. In this process, agricultural products are processed to generate added value. And in Ethiopia, the strategy for growing the agro-processing industry is through the development of integrated agro-industrial parks, or IAIPs. So the government has planned to establish 20 integrated agro-industrial parks. So far, 11 are operational. Most of the integrated agro-industrial parks process garments, some of them process textiles, some of them are specifically for agro-processing, some process mixed industrial processes, and a few of them focus on. The integrated agro-industrial parks follow a business model, and this business model involves what we call as the Rural Transformation Center. This transformation center provides linkage for supply of raw materials from the community, from the rural community to the park, and also serve the rural community in different activities. So farmers deliver their produce to the Rural Transformation Center. The farmers also receive agricultural inputs, social and other financial services. And these RTCs are sites for storage and primary processing of the agricultural products before they get transported into the industrial parks. Out of the 11 operational industrial parks, 4 of them are located in Addis within the city, and a few more are located around Addis. In recent years, there are programs to encourage urban agriculture in Addis Ababa. In most cases, the industrial parks and the urban agricultural sites are in close proximity to each other and to Agaki River catchment areas where the urban farming uses for microdam irrigation. Therefore, due attention is required in proper wastewater management of the industrial parks so that the rivers that are used for irrigation will not get more polluted. One of the agro-processing sectors is the leather industry, and this industry was the second foreign exchange earner in Ethiopia, amounting up to 98.4 million USD per year. The wastewater management system for the tanning industry is not satisfactory. In some cases, tanneries are strategically established nearby rivers so that the wastewater can be channeled without proper treatment and sometimes without even treatment. And this is an example of what you see when direct channeling takes place. And to the right side, You will also see the polluted water when it joins the nearby river. This is an example of a polluted river in Addis Ababa. Apart from agricultural activities, people still use this river for personal purposes, and that has its own health impact on the residents. In this talk, I will focus on two tanneries that our research group worked on. I will talk about the observations on the biological wastewater treatment systems that are in place in these tanning industries. I will also discuss on the microbial community composition, the potential pathogens, how they behave in these treatment systems, antimicrobial resistant bacteria and resistance genes, and the fate of this resistance in these biological systems. The first one is Mojo Tannery, which is located 75 kilometers out of Addis, the capital city. The treatment system is as displayed on this slide. It has the sequencing batch reactor as a biological reactor, which is followed by the constructed wetland system that has the emerging plant named Pragmitis. And when we look at the general performance of the treatment system, the removal efficiency of the COD, the BOD, the total nitrogen, and the ammonia are within the limit, and the sulfate also. The reason for having this integrated system is because tannery wastewater is highly loaded with nitrogen, different nitrogenous pollutants. So it is important to have a plant-based treatment system to have a complete removal of nitrogen. The effluent from the Mojo Tannery is received by Mojo River, which, as you see on the slide, joins an artificial lake, Lake Oka, that also is used for irrigation purposes for the production of different vegetables. When we looked at the microbial community of the integrated system using Illumina-based shortcut sequencing, we observed different groups of bacteria that were dominant at the different part of the treatment system. For example, the anaerobic part of the SBR was dominated by members of the group Delta Proteobacteria, Clostridia, Flavobacteria, and Sphingobacteria, whereas the aerobic group were dominated by members of the group Bacilli, Dinococci, and Beta Proteobacteria. And a number of diverse microbial groups inhabited the different parts of the wetland. In the process of laser making, there are different chemicals that are used throughout, and some of the chemicals are known as returning chemicals, xenobiotic chemicals that are very difficult for degradation. So using the SBR system at the bench scale level, we tried to biomonitor the indigenous microorganisms and the performance of these microorganisms with respect to the returning chemical degradation. And we observed that there are a number of microorganisms that are heavily involved in the degradation of the returning chemicals. With the help of flow cytometry and molecular fingerprinting, we have found that members of the group Proteobacteria, Bacteroidetes, Rhodocycales, and Caulobacteria were the dominant ones in terms of the degradation of returning chemicals. Also tried to see the fate of potential pathogenic sequences in the treatment system. And here we have presented examples of potential pathogens such as Shawonella and Clostridium, among the others. And the x-axis on this graph shows the different components, the SPR, anaerobic and the aerobic parts, and the consequent wetland sites. And the y-axis shows the percent abundance. And overall, a decreasing trend was observed for all of the potential pathogenic sequences across the treatment system. Different types of antibiotic resistance genes were found in this treatment system. And when we see the persistence, very few of them did not persist across the treatment system. For example, here we have the AADA resistance gene, which is a plasmid-encoded aminoglycoside resistance. So it did not persist, and we were not able to recover this gene in the effluent. But the other resistant genes, along with the majority of the resistant genes, were recovered from the effluent water. The second tannery is known as Batu Tannery, which is located within Addis. And the treatment system uses aeration, which is followed by chemical coagulation. Looking at the general removal efficiency of the different parameters, the system is not considered to be working in its fullest potential. Although the performance of the treatment system is not up to the standard, we try to see the microbial content of the influent water, the aeration system, and also the effluent water. And we see no differentiation in terms of the microbial structure. So what we see in the influent water is also seen in the aeration system, also in the effluent, and that shows that the maturity of the microbial group is yet to be worked on. The major microbial genera here also is the Clostridium group, and similar, that is what we also saw in the previous tannery, Mojo Tannery. When we look at the potential pathogens and how the abundance look like in the influent water, the aeration system, and also the effluent water. Most of the potential pathogens showed a decreasing trend, except for Clostridium, which is really important because this indicates that members of the genus Clostridium are carried out with the effluent water to the nearby river, which in turn will also be used for irrigation purpose. In a different study, we also tried to look at the antimicrobial resistance index of the Akaki River after the tannery provides the effluents. And what we have seen is that the AMR index with regard to different bacterial species is high in that particular river sites. So there are multi-drug resistant Enterobacter aerogens groups and Escherichia coli, and also Salmonella species. And that has its own impact because the Akaki River is being used for irrigation of different agricultural fresh produces that is supplied to the city. So having such a high antimicrobial resistant bacterial groups is a threat for the public health. So in conclusion, it's important to monitor microbial ecology of biological wastewater treatment plants for pathogen containment within the treatment system. And what we have observed is, at least in our setting, the constructed wetland system is convenient in terms of pollution, the high-strength tannery wastewater, both the priority pollutants as well as pathogens, potential pathogens. But work has to be done more on the antibiotic resistance genes because we have observed persistent throughout the integrated system. So perhaps it's important to optimize the integrated system for containment of the antibiotic resistance genes as well. Finally, I would like to acknowledge Professor Nancy La from the University of Michigan for the work done in Batu Tannery with the help of our PhD student, Dr. Tesfaye Admasu. I also would like to thank Addis Ababa University, the Biosciences in Eastern and Central Africa, ILRIHAB, as well as the UFZ, Germany, for the different laboratory activities done at different times. Thank you very much.
Thank you, Adi. You've seen you're online now. You managed to get through. If you want, you can switch on the camera, then people can also see you. Are there questions for Adi on her presentation? Yeah, she's registered here on the as being present, but so Adi, are you present? Okay, so Adi has chatted that she cannot join live. Then everyone who likes to interact with her, send her a question in the chat box or in the Q&A box on her presentation, so she can answer whatever you want to hear. And we'll have a short roundtable at the end. And I hope that in the meantime, that Adi is able to join also the online and not only the chat box. Then also as the view on time, we move to the next set of presentations, and Sergio will handle the chairing of that.
Yes, we are going to start with a presentation by Peter Naylor from Muharraq and Wastewater Services. And the title of the presentation is Design and Operation of SBRs in a Gulf Environment, the Reliable Production of High Quality Irrigation Water. And are you here to share your screen? Peter?
Can you hear me?
Yes.
I'm here, yeah. Can you see me? Can you hear me?
Yes, yes. Can you share the screen?
Sure. I will try with the technology. Excuse me a moment. Excuse the sunshine. Do you have the screen?
Yeah, but we see you, the webcam.
Yes, we don't see the presentation.
I'm going to see if I can... Okay, guys, I'm in difficulty with this. Apologies. Perhaps it's better if you play my video and I answer questions at the end. Would that be okay?
Yes, maybe. Thank you.
Okay, thank you. Good afternoon, everyone. My thanks to IWA for the opportunity to present today.
Eric,
Good afternoon, everyone. My thanks to IWA for the opportunity to present today. My name is Pete Naylor, and I'm a PhD student at Newcastle University in the UK. I'm carrying out research on the nitrification of wastewater at high temperatures and the challenges faced by large wastewater treatment works in the Middle East when trying to meet contractual performance standards. The title of my presentation is The Design and Operation of Sequential Batch Reactors in a Gulf Environment and the Reliable Production of a High-Quality Irrigation Water. The continuing privatization of the design, financing and operation of wastewater treatment plants, particularly in the Middle East, has resulted in the adoption of highly complex contractual arrangements between governments and international consortia. These parties are tasked with delivering the required services, often by way of public-private partnerships, or PPPs. They often comprise multinational finance houses, large international contractors, and water utility companies. Long-term concessions are granted to these consortia, often up to 30 years, and during this operating period, measures of performance are numerous and often determined on a daily basis. Failure to meet the performance standards can result in financial penalties of tens of thousands of euro per day and can be far greater than the revenue. There is therefore considerable onus on the designers and operators to ensure that the plant can deliver the required performance standards both on a consistent basis and throughout the year. Since my involvement with the scheme over the past 8 years, it's become evident that despite the plant operating conditions adhering to industry norms, there's been challenges in achieving compliance during the summer months for 2 parameters in particular, nitrite and turbidity. These observations have prompted this research. Accordingly, I am focusing on 3 areas: the analysis of historic plant data, laboratory-based nitrification assessments, and the analysis of activated sludge biomass by molecular tools. The treatment plant in this research is a conventional nutrient removal sequential batch reactor, operating on a 6-hour cycle with a typical aeration period between 2 and 3 hours. Process control is typical of industry norms in terms of FM ratio, sludge age, and dissolved oxygen. Analysis of plant performance data can be seen in the table and indicates that the plant consistently meets the required performance standards and when wastewater temperatures are below 35 degrees. These data, over a period of 3 years from 2018 to 2020, are generated by the operating company and clearly show the compliance. However, as temperatures rise, which is shown here in the top left graph, turbidity can be negatively affected. It's known that above 32 degrees, both the biology and the physical integrity of activated sludge flocks can be negatively affected. This is exacerbated by the need for increased aeration and therefore increased turbulence as a function of reduced oxygen solubility at high temperatures. As a consequence of this, the turbidity can increase, risking compliance. As can be seen in the bottom right graph, there appears to be a reasonable correlation between turbidity and wastewater temperature. Furthermore, the impact of nitrification is also clear, as demonstrated in the top left graph, whilst the oxidation of ammonia appears unaffected by high temperatures. These 3 graphs depict the loss and recovery of nitrate oxidation. We can see how the effluent nitrate increases during the hotter months, with a pronounced peak over 1 milligram per litre in the top graph. And the concentration does not return to pre-summer values until later in the year, approximately 0.2 milligrams per litre, even when the sludge age is increased to above 12 days, indicating a difference between AOB and NOB kinetics. As the wastewater temperature increases above 35 degrees, the loss of the ability of the plant to oxidize nitrite is clear, as shown in the more detailed graph on the bottom right. Indeed, the graph bottom left depicts a strong correlation between the recovery of nitrate oxidation and reducing wastewater temperatures. Regular assessments of the rate of nitrification by the plant biomass are carried out routinely. This will provide data on the kinetics of nitrification and on the changes in the observed rates throughout the year, as can be seen repeatedly in the graphs shown. Work has shown that the oxidation of ammonia and nitrite are zero order with respect to concentration, as expected. The implied Michaelis-Menten K coefficient for nitrate oxidation is considerably lower, however, than predicted values, published values, and below 0.3 milligrams per litre, and also appears unaffected by temperature. In addition, the minimum nitrate concentration achievable is lower than published values of 25 degrees and also less than 0.1 milligrams per litre of nitrite. Nevertheless, the rate of nitrate oxidation reduces from spring to summer, from May to July, as determined every 2 weeks, as the graph shows, indicating washout or possible change in the NOB population and supports full-scale plant observations. Later this year and into 2022, biomass samples from the plant from 2018 onwards will be analysed by published qPCR Amplicon techniques. This work will shed light not only on the phenology of the species forming the NOB population, but also whether the recently discovered Coromox bacterium is present. This analysis will hopefully assist in the selection of a more appropriate sludge age to be adopted to reduce the risk of non-compliance with nitrite parameter. In summary, it's hoped that the output from this work will add to the understanding of wastewater nitrification at high temperatures and support contract negotiators and design engineers with their endeavours. Thank you for listening. Happy to take any questions.
And thank you for the presentation. So, is there any question in the audience here? Mark.
Okay, thank you for a presentation and interesting data from the plant.
You're welcome.
Yeah, I have a question. So you have seen that the affinity constant you measured for the nitrite oxidation has no temperature effect.
Well, based on the views that we've got, yes, that appears to be the case.
Yeah, so based on your measurement, yeah?
Yeah.
Yes. Is this due to the fact that your K value essentially represents the diffusion into the flux you have, that the flux size is not changing with function of temperature?
Good question.
Or do you have a really suspended, really single cell culture?
No, we've got what you class as a typical activated sludge flow. What we do see in the summer, particularly, is that we get a probable breakdown of the flock because of the high temperature and because of the increased vigorousness of the aeration to achieve the dissolved oxygen concentrations required. Because you're looking at 35, 37 degrees, oxygen solubility is quite low. So you've got to inject a hell of a lot of air to reach 2 milligrams per litre of DO. So I think it's a function of the reduced stability of the flock because of high temperatures and because the more vigorous action of the aeration process to try and get dissolved oxygen into solution. So it may well be that you've got a smaller flock size at high temperatures. We haven't looked at that, to be honest, in great detail. I suspect it's a gradually reducing NOB population with increasing temperature. But so we're looking at doing some qPCR work later this year, and hopefully we'll be able to quantify whether there's a change in the NOB numbers and possibly change NOB species as the year goes on.
Yeah, thank you. Would indeed be interesting to see, and I fully agree that it's the washing out of the NOB, and it would be interesting to see if
it probably. I mean, certainly at 30 degrees in January, February, we have no problem whatsoever, and we're running about a sludge age about 7 days, which is you could argue if you sit and crunch the numbers in terms of design manuals, that's still relatively long. But we don't have a problem at all at 7 days, and we tend to increase the sludge age not until about April or May as the temperature rises. But even then, we're only talking maybe 12 to 15 days. But it may be that's not early enough. It may be we have to increase sludge age to maybe 12, 15 days, perhaps as early as March, when the temperature is only maybe 32 or 33. But until we look at the individual species and the numbers quantitatively by PCR later this year, we're not going to know.
Just the last detail, in what time frame is the temperature increasing? Is this within, certainly in 1 or 2 weeks, or is this over a few months?
Oh, it's over months. I mean, it's a gradual increase from maybe 27, 28 degrees in December, January, to around 35, 36 in July and August.
Yes,
but the actual the impact on NOBs is not seen till about September, October, because we're still the plant's still 35 degrees now, and we're at the end of October, because you have such a long time for the plant wastewater temperature to reduce from 35. It's not until November we get down below about 32 or about 33 or 34.
Thank you. Interesting, and like to see the data later on.
You're welcome. Thank you very much.
Thank you. Well, if more people have more questions, you can write it in the Q&A, and then we will send it to him. So we are going to move to the next presenter, Eleanor Frank from Sweden Water Research, that will present using knowledge of bacterial communities in wastewater to assess pollution of bathing water and marine sediments. And we will launch now the video. We are having a problem with the video. It says that it doesn't exist. Eleanor, are you in the call? Maybe you want to present live?
The presentation via email.
We can move to the next one, and then we'll try to solve it meanwhile. So we will have the next presentation by Kwanrawi Sirikantana from Chulaphorn Research Institute, and the presentation is about antibiotic resistance gene abundance and the correlations with fecal source tracking markers at tropical recreational beaches. And we will launch the video now.
Hello, everyone. Thank you for coming to my presentation. It is my great pleasure to share with you the work that I'm doing on antibiotic-resistant genes abundance and the correlations with fecal source tracking markers at tropical recreational beaches. In this presentation, I will divide my presentation into 4 subsections. First, I will introduce you to the situation of antimicrobial resistance and coastal water pollution in Thailand. Thailand is a country in Southeast Asia with tropical climate. Our latest annual report of coastal water quality is shown in the map, with the orange color as poor water quality and the yellow color as the fair water quality. The beach water can be contaminated by activities near the beaches, for example, the food stalls, hotels, restaurants, and combined sewer overflows. We have the Thailand National Strategic Plan on Antimicrobial Resistance 2017 to 2021. And this is in coherence with the Global Action Plan on the Antimicrobial Resistance by the World Health Organization. The National Strategic Plan is guided by the One Health concept with interactions between three sectors: human, animal, and environment. However, information from monitoring and transmission of AMR in the environmental sector in Thailand is still limited. Therefore, the work that I'm presenting here will provide the insight into the ARG pollution status of Thailand tropical coastal waters and could facilitate effective management policies to minimize the ARG dissemination in marine environment. So in this project, we monitored antibiotic resistant genes and microbial source tracking markers for human sewage. Our study sites are 2 recreational beaches located in eastern Thailand. Beach BS served over 5.7 million visitors, mostly were the Thai people, and Beach PT was visited by 31 million people, and mostly were the foreigners. Both beaches have a history of 5 to 35% exceedance of the bacterial water quality standards. In this project, we sampled 42 samples from 3 stations from the Beach BS and 4 stations from Beach PT, and samples were collected for 3 times in the dry season and another 3 times in the wet season. The sample collection was conducted on Sundays at waist depth to mimic swimmers' exposure to beach water. The sampling started at 9 a.m. to avoid strong sunlight, as this can affect microbial survival in water. 1.5 liter water samples were pH adjusted and filtered. Then DNA was extracted and the qPCR was performed. The first objective of this study was to investigate the spatial and temporal distribution of a class 1 integron integrase gene, INTI1, and ARG is conferring resistance to commonly used antibiotic in Thailand, comprising BLA-NDM, BLA-TEM, BLA-VIM, MCR1, SAL1, and TEDQ. The second objective was to assess correlations between the ARG, INTI1, and the Mst markers, human polyomaviruses, HPYV, and crassphage, which have been validated in Thailand, and they show the high specificity and high sensitivities. We have the hypothesis of a portion of ARG pollution originated from human sewage, and therefore human-specific Mst marker could serve as an indicator of ARG pollution. Next, I will show you the results of this study. For part 1, the spatial and temporal distributions of ARGs at 2 beaches were presented. We found that BLA-TEM, INTI1, SAL1, and TEDQ were dominant, and the INTI1 level was the highest, agreeing with other studies. This study reported the first BLA-VIM in marine water in Thailand, which was found mostly at Beach BS. MCR1 was not found in any of the samples, while BLA-NDM was detected only in the Beach BS samples. BLA-VIM, BLA-NDM, and MCR1 genes were all found in the influent and effluent of the municipal wastewater treatment facilities near both beaches. These are from our unpublished data, and this imply a dilution effect of the genes in environmental water from the wastewater sources. We also found that temporal and not spatial variability was observed for ARGs at each beach. Blood temp was higher in the wet season. This suggests a sentinel site for water quality monitoring, as well as a higher sampling frequency, especially during the wet season. For part 2, there are correlations analysis among the ARGs. First, INTI1 correlated with SAL1 and TET-Q at both beaches, and this suggests potential horizontal gene transfer as the main mechanism driving the ARG diversity. SAL and TET genes were also correlated at beach BS, and this may imply that they originated from the same pollution sources because co-presence of both genes were reported in wastewater treatment plants, aquaculture, and livestock farms. Moreover, cross-selection of these two antibiotics to the two ARGs has also been reported. For part 3, we looked more into correlations between ARGs and MST markers. We found that crayfish correlated with INTI1, SAL1, and TET-Q at both beaches. And because both crayfish and HPYVs have been validated for their high performance, meaning that they are very specific, very sensitive as human sewage indicators in Thailand. Also, crayfish was significantly higher than the HPYVs in human sewage and tropical freshwater, but lower in the seawater. This all suggests that crayfish has a higher decay rate than the HPYVs in seawater. Due to the explicit association between the concentrations of the crayfish and some of the ARGs, the ARG occurrence in the samples were further investigated in relation to the crayfish levels. These figures show crayfish concentrations here. In samples with numbers of co-occurring ARGs. The medians are shown in solid lines. Dark circles indicate quantifiable crayfish results, and white circles indicate the data below the method limit of quantification. So we found that higher number of higher number of ARGs tended to co-occur in samples with higher crayfish levels. This implied that sewage is one source contributing to the marine water pollution. In samples with no detectable crayfish, there were still at least 4 and 2 co-occurring ARGs at both beaches. And these results indicate that there could be background levels of ARGs present in the beach water. And concentrations of ARGs were enhanced by human sewage input as indicated by the crayfish marker. But other confounding factors should also be considered, including different decay rates and laboratory recoveries. To conclude this study, the study reported that blood temp, INTI1, SAL1, and TET-Q were dominant in 2 tropical beaches in Thailand. Temporal variability was observed for ARG at each beach, suggesting that we should have a sentinel site for each beach and higher sampling frequency, especially during the wet season. Crayfish can be a proxy of ARGs due to the correlations with some of the ARGs. And higher numbers of ARGs tended to co-occur in samples with higher crayfish concentrations, implying that sewage contribution to the marine water with a persisting ARG background. So before I finish, I would like to acknowledge the funding sources from Wolailak University and Jelapun Research Institute. And thank you all for your attention.
Thank you. Is there any question online or here on site?
not, then I've at least one question. Nice presentation. The grass phage is nicely correlating with the antibiotic resistance genes, and you make this equivalent to that they come from sewage. How are you sure it's coming from sewage and not from bathers?
Yes, thank you for your questions. So we've been testing in our previous studies, we tested the specificities of the grass phage with the human sewage from the populations in Thailand, and it does not cross-react with other animal feces. But your question about the bathers, right? So that's another interesting point as well. We haven't really tested the grass phage with the individual feces yet, because we think that for our pollution problems in Thailand is more from the sewage, from the leakage sewer line, or from runoff, or for something like that. So from I guess that from your question, I cannot be sure exactly that it's come from the bathers that come to play in the water right at that time, or it's like from the wastewater contamination that's already been there. But another one thing that I can tell you is that we tested 2 different human-specific markers, the grass phage and human polyomaviruses. Both of them are equally specific and sensitive, but somehow we found human polyomaviruses at higher frequency and at higher abundance in the seawater than in the than grass phage. So this thing is still like it's still a little complicated, but I think we will have to study more.
Oh, thank you. It was also because the beaches were heavily used, but maybe time dynamics, if you can get that data over time, might indicate what is the source. But thank you for your answer.
Thank you.
Thank you. Is there any more questions in the room? Well, again, if anyone online has questions, you can write it in the Q&A. So then we are gonna go back to the previous presentation that we found back the video by Eleanor Frank. So once we share the screen, we can press play.
Hi, my name is Eleanor Frank, and today I'll be talking about my research project in which we're using knowledge of bacterial communities in wastewater to assess pollution of bathing water and marine sediments. I'm employed at Sweden Water Research, and I'm working at the Faculty of Engineering at Lund University. I would like to mention FOI, here's the Swedish Defense Research Agency, for their collaboration with DNA sequencing and source tracking. And also a big thank you to the city of Malmö and the city of Helsingborg for their funding and involvement in this project. The sample collecting was performed from a boat, and by lowering this sample core sampler down to the bottom. And from this, the top centimeter was collected into a sterile 50 ml Falcon tube. So where were these sediments taken? Here's Europe, and here's southern Sweden, and Öresund, which is a narrow strait between Sweden and Denmark. And the samples were collected just outside of Helsingborg, at the narrowest point of Öresund. The origin of the project was in summer of 2018, which was very dry and very little rain. And this resulted in very little combined sewer overflows in Helsingborg, but they still had massive problems with E. coli at their beaches. So the question emerged, where did these E. coli come from? During March 2019, the first sediments were retrieved by boat. What you see here is the locations of the samples, as well as their Code Alert 18 results. And this point here, 1250, is the sediment sample of
Put a bit back and start again.
18 results. And this point here, 12.
Put it back.
It seems that we are having problems again with the video. It
Massive
stops at a
problems with
certain
E.
point.
coli at their beaches. Defense Research Agency.
Do people have the same problem if they play the video themselves at home? Yeah. So we are gonna try to solve it again. So yeah, let's move meanwhile to the next presenter, and that is Alessio Cavallaro from IVAG, that will present Legionella species abundance in shower whose biofilms correlates with specific microbial communities, exploring potential molecular indicators, and will present, I think will present live. Are you here?
Can you hear me?
Yes, you
Oh,
can share your screen.
Good. Just a second. Can you see my presentation?
Yes, we can see it.
Great. So good afternoon, everyone. My name is Alessio Cavallaro, and I'm a PhD student at EAVAG and ETH Zurich in Switzerland. And today I'm going to be talking about my work on how microbiome composition correlates with the presence of Legionella in shower hoses biofilm. So, all right. As probably many of you already know, Legionella are human opportunistic pathogens, and they are etiological agent of the Legionnaire's disease. They are usually found to be living in biofilm where they engage all sorts of other interactions with the organisms, and some of these being positive, for example, with protozoa, where allowing their replication. Some of these can be negative, like certain forms of antagonism. Now, what we are really interested in is understanding more about these interactions that Legionella engages in biofilm to gain a better understanding of their ecology. So in particular, what we did to explore the ecology of Legionella, so we started from a building with a known history of Legionella contamination. From this one building, we collected 100 shower hoses, and we extracted the biofilm out of the shower hoses to perform DNA extraction that was used afterwards for downstream analysis. In particular, we performed 16S and 18S Amplicon sequencing to characterize the microbial and eukaryotic community. And we performed digital droplet PCR to quantify Legionella species, and specifically Legionella pneumophilia, which is the main strain related to the disease among the genus Legionella. We used the data afterwards in order to explore potential correlations between Legionella and other microbiome members. Now, just for you to have a look at what we did in the lab, this is how we performed the biofilm extraction. So we started from shower hoses, and after putting glass beads and water inside, we sonicated in order to make the biofilm detach. And this is what a eluted biofilm look like. And you can see this in this beautiful picture in the middle of the slide. Now, I must say this was a clean sample. I've seen worse. But so the DNA was extracted. So this is the biofilm that we used to extract the DNA that was used for further analysis. Now, what is really interesting about the data that we generated is that despite these shower hoses were similar in age, material, and user profile, and I want to remind you they were coming from the same building, we noticed several differences in relative abundance among the samples we collected. For Legionella species, this was ranging from 0 to 5% relative abundance, and for Legionella pneumophila, this were ranging from 0 to 0.16%. And this kind of led us to the hypothesis that differences in this Legionella presence among our samples were somehow driven by the overall microbial composition of the samples. If we look at the general ecology of a shower hose, we can see how among several other organisms which compose a very beautiful, diverse samples community, we see organisms that were previously already defined to be living in water and biofilms, and we notice how Legionella among our samples was one of the dominant genus. Now, moving on to the actual analysis, what we did was applying different statistical methods in order to explore potential correlations. And so, in this slide, you can see on the left-hand side a random forest analysis, which is a regression method based on decision trees, that we applied on the extremes of our distribution of relative abundances, picking the high Legionella samples and the very low Legionella samples. And we run the classification random forest to find correlating organisms, which are displayed here in decreasing importance from Aquabacterium, which, for example, correlates with low Legionella abundance, to other genuses correlating mostly with high Legionella abundance, and a few of them also correlating with low Legionella abundance. Now, all these results were backed up by other statistical analysis, like these linear Spearman correlations that you can observe here. So this is how a negative correlation look like, while this is how a positive correlation look like. And so we kind of can say that some organisms really do correlate with high or low Legionella abundance in our samples. And this is just an example, but we performed this using the eukaryotic community. We performed this both on Legionella species and Legionella pneumophila in order to find the most broad correlation range that we can. So this is a nice example of how from a complicated community where thousands of taxa are correlating one to the other, we can actually focus on the organisms that we are interested in order to find correlations that can be used for different kinds of application. And I want to talk about this in a minute. But so in conclusion, we can say that we were able to identify positive and negative correlations between Legionella and other microbiome members in shower hose biofilm samples. But of course, this was just one study, and we are aware that additional studies are necessary in order to confirm this preliminary observation and maybe to find universal trends, and so to find real molecular indicators of the presence or absence of Legionella in a given sample. And because we are also aware that correlation is not causation, and that taxonomy could have maybe not could maybe provide not enough information about what these correlations actually mean, probably function-based metagenomic data could provide more sensible information than taxonomy-based correlations. And we are working on that in order to perform the necessary steps to confirm what we are observing with these samples. And allow me to share with you now what I'm trying to do during my PhD that I started 1 year ago. So the final goal would be to find a possible probiotic approach against Legionella in plumbing systems. Now, this can be approached by two sides, and one is the one that I just now described to you. So looking at sequencing data from real samples, statistical correlations, and find the organisms that are correlating with the absence of Legionella, and maybe using them for to create a probiotic community. The other one is what I'm also doing on the side in the lab, where I'm isolating environmental bacteria, randomly isolating them and testing them for their ability to inhibit Legionella on agar plates. And eventually, I want to move from agar to more real system, and possibly connecting the two sides in order to find the best, or in order to explore if this is even feasible to create a probiotic community that can destruct Legionella, that can fight Legionella in plumbing systems. Allow me at the end to thank our project partners that make this possible, and the great group I'm part of at EAVAC in Switzerland, the Drinking Water Microbiology group. And of course, thank you for your attention as well.
Thank you, Alessio, for the presentation. Is there any question from the audience, online or on site? And I can ask you something like about your project in general.
Yeah.
This thing that you said about the probiotics for for the Legionella treatment, like how could you apply that in real systems in the plumbing, like?
Yeah, that is a great question. So I have not clear in my mind what a final product would look like, but of course there are some aspects that one have to take into account. And for example, being these, all the organisms that are used in a probiotic community must be organisms that are otherwise not pathogenic, of course. They have to be organisms that can attach and survive in biofilms, because otherwise they would be flushed away in plumbing systems. Of course, they have to kind of be resilient in there. So I don't know how a final product would look like, but I have clear in my mind what kind of necessary how can I say like what are the necessary things that a probiotic community should have in order to be able to be applied.
Thank you. We also we have a question here in the room by David.
It's a follow-up on this discussion that you had.
Yeah.
So when you speak about probiotics, so you mean that you want to inject a chemical compound that would stimulate the other organisms? And what happens if it ends up in the drinking water that we drink in the end? Is there an effect on the gut microbiome as well?
So honestly, I was more thinking about actually shaping the microbial community via colonizing the plumbing systems with other organisms that can have a negative effect on Legionella. So more that than injecting chemical compounds. And of course, for this to be possible, all these organisms must have not a negative effect on human health primarily, because this is where the water would be consumed and would be drunk. So this must be important.
Because if you want to stimulate some microorganism to be in the system, you need to provide some substrate to stimulate their growth. So it means at a certain point you had to add something chemically, I assume. Yeah. Thank you.
Yeah. Thank you.
And we also have another question here from Francesc.
Hello, Alessio.
Hi.
Assuming the relation between certain microbial species and Legionella is indeed there's really a direct cause between them, do you assume, or what would you guys? Is it a taxonomy-based relation, or is it more a functional one? I know you said you were going to check it, but what is your initial guess?
My initial guess is this is more function-based. Of course, first of all, with the 16S sequencing, we cannot go most of the time very deep in taxonomy, and so we cannot go down to the species level. And one genus within one genus, we have multiple strains and multiple bacteria, and this can have different functions. So my guess is this is more function-based, and I would like to explore that, and we are actually in the process to do that with more metagenomic-based approach. And once we have a better understanding of what are the functions, and if there are functions that correlate with the presence of absence of Legionella, I think we will have a better understanding of how this ecology is actually working.
Could you give an example of which functionality could prevent the growth of Legionella? And is there something in literature already?
Yeah, for example, we know that Legionella has very unique nutrient requirements. So it's an auxotroph for 7 amino acids, and it needs iron to survive. So, for example, if in the community there are organisms that are greedy for iron, and they have mechanism to scavenge the iron from the environment, and I'm thinking, for example, siderophores or something like that, this could have an impact on the way Legionella grows. And so we would be maybe able to reduce the growth rate of Legionella by just attacking what are the nutrient requirements, or if we know that there are antagonistic behaviors like toxins or something like that, we could more target Legionella growth in plant systems.
Thank you. And one last question that I didn't see, sorry, in the chat before, by Dr. Amit Pinto. Could it just be a matter of chance that Legionella are in some samples and not other?
It could be a matter of chance, but because, as I said, the shower hoses that we collected were coming from the same building, and they had very similar characteristics like age, user profile, and material. I like to think that this was this is really driven by the overall microbial community. But of course, there's the chance that this is just random, and this is why we are trying to explore that more.
Thank you. Then you have some questions also in the chat that you can answer later.
Yeah, I will.
So let's move again to Eleanor. So try to share your screen to see if we can watch the video now. And I think we left it in minute 2 and something. Are you there? Yeah. Eleanor? Ah, yes. Can people hear? Because in the room we cannot hear the audio.
At the moment.
Just I try again.
You can't hear me? Oh, you can't hear the video?
Now we can hear you.
Now we can hear you, and I think at the end we could hear the...
You hear this?
We can hear it online. And now the voice is gone again.
Oh, okay, I don't know if it wasn't our problem or not. Yeah, we cannot hear the video.
They can't hear the video.
Otherwise, you could put the PowerPoint and present live if you want.
Screen, can you also select share sound? When sharing the video, also select share sound, so the sound also comes through. Click on the screen sharing button, and then you also have to click on share sound. You should see the share sound button. just click on share screen, and then you should see, yeah, click on that. And then you should see share sound in the bottom left corner. The button is called Share System Audio. It should display in the bottom left corner of the screen sharing pop-up.
And
the problem
can you
is that
send us the video again
the
to see?
video that you sent us. Pardon.
Or can you present live if you share the PowerPoint? Well, while we try to solve this again, we are gonna move to the other presentation that it's by Zore Kurt from Middle East Technical University about an education of rural communities of Colón, Panama to treat and keep their drinking water sources. And this
I'm
was
here. Thank you very much.
a live presentation, but I don't see her in the call, so we will.
No, I'm here. Can you hear me?
Yes.
Yeah, I'm present.
I didn't see you. Sorry. So then, yes. So you can share your screen.
Of course, no problem. Why does it tell me to? Can you see my screen?
Your screen with your initials.
No, we don't see the sharing screen.
Yeah, because it asked me for something.
Maybe
don't
on
see
the
my
left, sometimes you have to give permission, or.
It asks me for video link, but I actually want to share my screen. Can't I just share my PowerPoint? Sorry about that, but.
Yes, if you click the third icon that says stop sharing your screen, then you can.
Exactly, that's what I am doing. And when I click it, it says share video, and then it says video link. I have no idea why it does that.
When you click, don't you have the entire screen option?
No, I don't. Unfortunately. But I do have a YouTube video link for my talk, but I much rather talk to you here when everybody's present. I think I might have managed it. Just a second. Select window, screen, entire screen. I think I got it. Sorry, it's just Zoom has been the thing that I was working a lot on.
Otherwise, we can launch the video, and then for the questions you are.
Either or is fine by me, honestly.
Okay,
I think I think I got it. Didn't I?
Yes. So now, now we see your presentation.
Okay, perfect.
So you can put full screen and then.
Sorry. Yep. Now I did it. Well, sorry very much. Hello, today we will talk about something quite different. I've heard many of your talks. You were talking about more sophisticated things as identifying species and metagenomics. However, even though we would have liked to do some similar analysis here in Panama, we unfortunately don't have that much sources. I'm from Turkey, but I do work in many projects in Panama, and we work in Colón area. I want to first start with talking where we chose this area to begin with. As you might already know, Panama is in the middle of southern North America. It has this huge canal. But one thing that Panama is not known with is the communities that live in Panama, and then they have many indigenous communities. What you see here in this side of the Panama site, very close to where we chose, is a land that is dedicated to indigenous communities. That means that no water or wastewater resources are delivered to that community, and you actually are not really allowed to be in there. That is why we were not in the indigenous community, but we were in a community that is sharing the same water sources with the indigenous community that, again, don't have access to any type of treatment of water or wastewater. However, they are supposed to provide their own drinking water with their own means. And considering the fact that those communities are actually poor communities and are trying to develop, there are two problems that we're seeing in those communities. One is the quality of the water that is not known, which they actually get from the wells that are not really engineered. So they drill wells on their own randomly, and they get the water from there. And another problem is that they don't have a mean to be able to get the quality or how to use this water, or they don't know how to treat it if there is any kind of a problem. Also, those communities have been developed, which means many people from the rural areas of Panama are trying to move there because it's a cheaper place to live, and uncontrolled pesticides and fertilizers have been used in the area. Past in the day, several pesticides like atrazine and 2,4-D have been analyzed in those water sources, and that was the reason why we picked the area, why we picked it. Like I described to you before, water is a huge problem in the communities. What you see here is a well that is actually gifted to the community by the industry that is working nearby. They have huge open sea fishery farms that belong to US that are in that community, and just to help out, and also because they have people that work in those industries and live in that area, they sometimes do drill 1 or 2 wells, and this is one of the wells that is providing the water to a community that is around 110 families. And this is really our area of interest. I wanted to show you the sampling points that we did in this project. In this project, we have sampled many different communities, like all the time for about 2 years, estimated the water quality in the sources of the wells and also the rivers sometimes. The reason of sampling the rivers was because in some of the communities were near the open blue sea farm, where I had described you the problem, is because of the rainy and dry seasons. Sometimes the river is used as a water source rather than just the well itself. But of course, we have sampled the other areas around there. I want to quickly show you the water parameters, and this is not the entire data set, of course. This is like a selected data points that are representative. What we saw was that most of the time we didn't have any problems with the quality that we have analyzed. The pH was really like stable, conductivity and even hardness. What we saw occasionally was change in the salinity in the wells, and it corresponded to the dry and wet seasons. So whenever there was a dry season where there is not too much rain, we were observing that the water salinity in the wells were increasing, which we were relating to maybe a mixture from the ocean, because the wells are quite close to the sea level, which could have been a problem if over use of the water is present in the area, and of course can affect agriculture activities. Another item that we have seen most of the time is iron and copper, that is most of the time higher than what normal drinking water standards were, and it was changing, of course, from time to time. The randomness of this change, it didn't have a trend of the change. That's why we went to the water sources of the areas where we found that the iron and copper concentrations were higher. And what we saw was that the source didn't have any problems. The source was actually quite clean and very good in all the parameters. However, when we were going downstream of the river, or when we were going to like wells that were closer to the ocean or closer to the river, indicating that that might be a mixture of the water from the river with the aquifer maybe, or with the well, we were seeing the increase of copper concentration, which we thought could be related with the fertilizers. You don't see the fertilizers and pesticides. You don't see the data here, but we also have analyzed soil samples throughout the years. And what we saw is seasonally, sometimes we had a high increase in 2,4-D concentrations in the area. We were able to isolate bacteria that could degrade 2,4-D, atrazine, and imidazole. However, these pesticide concentrations were not kept in any dangerous concentrations or very high. That is why we actually focused on educating the community on keeping the water sources cleaner, and also teaching them some ways that we have designed for them to get rid of the iron and copper concentrations that were high. When we went to the households and we analyzed the water, what we saw is constantly high fecal concentrations in the waters in the houses. However, this concentration was not present in the in the sources. So first, what we did was we organized seminars of the communities to teach them how to do clarification. In the area, what you see here is a brochure that we have been distributing to the community, and that's why my title is education. We have also worked with the health centers and the schools to be able to provide that information. Then we gave a little seminars that they were like in total 4. We planned for more, but because of the pandemics, we were not able to get more than that. Where we were teaching the community to boil their water, keep them in closed containers. And of course, we were teaching them about filtration, because sometimes they will see sedimentation. And of course, how to keep the water in containers. What we have actually developed with this project was a homemade biochar. In the area, coconuts were really an entity that was present, and of course the shell and the outer shell and inner shell of the coconut is a waste. I'm not talking about the waste issue in the area, but that also is present. We have analyzed several different materials to use as water treatment means, and we just found that coconut is one of the best items that they can actually use a can of beans. We also taught to the community, this is actually an engineered reactor, but we've made it in a smaller scale, where coconut shells are being present in, and that can be converted to the biochar. What we did with this biochar was we analyzed the adsorption and desorption capacities of this biochar, inner and outer shell for copper and iron. What we have seen was that the biochar, of course, was not as effective as activated carbon. What was performing enough with the capacity present here that was sufficient enough to be able to treat the water that is coming to the community. And that is what we incorporated in our educational seminars for the community. As a result, we conducted surveys in the community and we actually collected the health data. I'm not allowed to share the health data because of the ethical issues, but I wanted to show you some examples of the survey, which people were believing that the seminars that were provided, the educational components that were provided with the project, actually were able to solve some of their problems, which they indicated as water. And of course, for the trash, we were not able to do anything yet. With that, we actually concluded the first part of our project, that was really educating the community and keeping up the water quality in the area. And we are planning to implement similar means in other parts of Panama. This was a project that was supported by the Panamanian government. Thank you very much for listening. And I think I need to cut it short because I just had 10 minutes. And thank you very much for listening to me. I'll be very glad to answer any questions if they're present.
Thanks a lot for the presentation. Indeed,
Thank you very much for having me.
We have a question from the online audience from Peter Naylor. Did you have any issues of people not wanting to add hypochlorite because of the impact on taste?
We did have people that didn't want to add hypochlorite because of its cost rather than the taste. The main problem of the community really is a financial issue. And that is another reason why we're suggesting them to add only 1 or 2 drops, and we haven't received any feedback that affected the taste of the water. We had some resistance of filtration. People didn't want to do filtration really in their houses. They thought that the water filtration was not really solving any kind of a problem, and they much rather wanted to have financial support to be able to get bottled water rather than using their own water in the community.
Thank you. And we have another question from Dr. Adele Lazuka. And thank you for your talk. How can people face salinity?
Face or taste?
Face. I think I sent the question from...
Everybody in the community is aware of the fact of the salinity. Nobody really complained about it. This was our measurement that we realized that salinity was changing over time in the wells. But like I said before, whenever the well water really is not coming with the pressure that it is coming, the communities most of the time mix it with the river water that is coming out there. And of course, what they're facing is a water that is coming with sediments from their faucets. And that is a problem. And the community members never really complained about salinity or anything else. They actually really want the government to provide some sort of like piping system for them to have treated water, but that is not going to happen anytime soon.
Thank you. We have another question here.
Of course.
Hello. First, congratulations.
Thank you very much. I much rather had a more sophisticated project, but I'm sorry that it's so primitive in your conference, whatever I presented.
I think it's fantastic, honestly. I have 2 questions. The first one is, I saw that you did not measure arsenic nor manganese in the groundwater, and they are 2 of the main contaminants of groundwater, at least in Europe and Asia. And second, how did you deal with trust and mistrust within the Panamanian communities?
Very great questions. For the first question, before we started the project, we actually did a very detailed analysis of 120 pesticides and I think 82 different categories, which included magnesium and arsenic. So what I showed you was the stuff that we thought actually might be affecting. So we did a first analysis in the community that I showed you at the very beginning where Open Blue was, and there was no problem with arsenic or magnesium. And actually in Panama area, that part, which is north of the Pacific, doesn't seem to have any arsenic issues, just like the south of it, because south of Panama has a different composition of the earth. So we knew that, and that's why we didn't measure it. As for the second question, that was the reason why I was not the one who was taking the samples, even though I really wanted to be able to go and do all the samples and do the surveys. A community that is close, that is small, that is not really well taken care of by the government, and has a huge population of a certain race, really doesn't trust a person who comes from outside, thinks that she knows what she's doing, and is not one of them. That is why I hired several people that were working in the community. What happens in Panama in that site is, like I showed you several maps with points, that huge area divides within itself to regions. Every region chooses one person who is responsible of certain items, including the water, and then they have a council. So I did hire 2 people from the council to be able to help me with talking to people, or even taking samples, or do the rest of the work to be able to really achieve the goals of the project. So it was not me who went to the houses to interview people or even take their samples, because they would not give me. Also, the main problem was that hidden wells. So people really thought that we will punish them. And that's why they were not giving the locations of the wells, but having like this person from the community and also having another assistant of mine that was there every week. After a year, they have gained trust and they showed us, maybe not all, I am not aware, but a few of the wells that they were using as a water source.
Maybe just a small comment for you, because you mentioned a few times that your methods were primitive or simple. For studies, it's not going about primitive, simple, or complex, but it's going about being ingenious and relevant. And your contribution is certainly on both factors high. And
Thank you
that's
so
how
much for the
contribution should be.
Thank you.
So, Francesc has more questions for you.
Okay, this.
Of course, that you is me.
Yes.
Okay. Okay.
There is. I asked this question because there's people in the group I work in, doing similar projects in Bangladesh, and they use the same strategy of hiring local people to increase the trust and transparency of the community. So it's nice to see that the same strategy works in other parts of the world. I have in mind a project that is taking place now in Ghana, in which they want to implement a similar study, a similar case, new technology, or trying to educate people on how to use the water they have. But they saw that the community was very reluctant, because what they said is that external people come, implement their technologies, regardless of whether they are simple or complex. The technology, after a couple of years of this technology working, it breaks, and there's no one with the expertise to fix it. Did you find the same problem there, or is this something that you think, can you anticipate that this is something that will happen there, or do you plan to educate the people there on how to fix the devices or the technology that you bring?
That's an excellent question. I thought of how we could actually continue convincing people to be able to be able to use whatever we provide them, because I feel like this is going to be a problem really in the community. I didn't think of having somebody there all the time who will be able to fix the technology, because I cannot have a project that is 20 years long to be able to support somebody there financially. What I actually wanted to achieve with this project was more of warning the government, so that at least they take some sort of a mean and tell them that this is really not something that complicated, that maybe they can dedicate somebody there to work with. And I understand your concern, and I still feel very sorry, once the project is totally completed, to leave the community there, because you also have sort of an attachment to the community and feels like you're leaving them on their own. And this also, of course, can be a repetitive behavior. And I do believe that is the main reason why the community doesn't want to trust anybody else, because what they have seen is that people come, they do their work, they think that the project really pays you a lot of money to be able to do that, and you're gaining everything and they're left alone. Another thing that we never spoke about was the community sometimes doesn't have only water issue. For example, the last month when I was there, they were telling me in their council meeting that even though water was a good thing, now they have problems with crime. So what are we going to do about it? And that's something that I really cannot do anything about. So there are so many points that we cannot predict. And unfortunately, as a scientist, probably I should have thought of a way to fix the technology and for them to be able to continue using it. But maybe we should work with some sociologists to be able to do a better work in the area.
Or maybe educating the people should be an integral part of these kind of projects in the future.
Yeah, exactly. That is the reason why I actually provided most of my educational materials to the schools, because I believe maybe kids are easier to mold, and also the health centers, because whenever people really are sick is when they understand that they have to be able to do that. That is also why all our seminars happened either in the schools or in the health centers. And those were the people that were in close touch, like the nurses and the teachers. And I thought those people are more sustainable and they will be there for a longer time. Of course, I have no evidence or no proof that they'll be there.
Thank you for the nice discussion, and maybe we can continue in a bit. Now we solved the problem with the video, so we are gonna play it again, and let's see if this time we don't have more issues. Sorry for the inconveniences. And I think David wants to say something.
We cannot hear you, David, if you're talking.
You can hear me?
Now, yes.
Yes? Okay. So thanks. I don't know why. It's Murphy's Law in the evening. So Eleanor, our apologies for this issue. I understand that you can be stressed, and there is no way that we will not pass your presentation. So we do our best to have uploaded it, and I think it works now. Maybe Sergio, you can help me to launch it. I think I just have to. No, I let you do it. And we will continue the next session. We will make a break and we continue the next session in 10 minutes with Colin and the other colleagues.
Hi, my name is Eleanor Frank, and today I'll be talking about my research project in which we're using knowledge of bacterial communities in wastewater to assess pollution of bathing water and marine sediments. I'm employed at Sweden Water Research, and I'm working at the Faculty of Engineering at Lund University. I would like to mention FOI. Here's the Swedish Defense Research Agency for their collaboration with DNA sequencing and source tracking. And also a big thank you to the city of Malmö and the city of Helsingborg for their funding and involvement in this project. The sample collecting was performed from a boat, and by lowering this sample core sampler down to the bottom. And from this, the top centimeter was collected into a sterile 50 ml Falcon tube. So where were these sediments taken? Here's Europe, and here's southern Sweden, and Öresund, which is a narrow strait between Sweden and Denmark. And the samples were collected just outside of Helsingborg at the narrowest point of Öresund. The origin of the project was in summer of 2018, which was very dry and very little rain. And this resulted in very little combined sewer overflows in Helsingborg, but they still had massive problems with E. coli at their beaches. So the question emerged, where did these E. coli come from? During March 2019, the first sediments were retrieved by boat. What you see here is the locations of the samples, as well as their ColiAlert 18 results. And this point here, 1250, is the sediment sample that was taken just by the treated effluent from the sewage treatment plant in Helsingborg. ColiAlert, however, was mostly used as an estimation of the problem. The main focus was to look at the V3-V4 region of the 16S rRNA gene to get the whole bacterial community of the sediments. And taxonomic assignments of these ASVs for data used Nmds database, which stands for Microbial Database for Activated Sludge. And here you see visualization of the variation in the data set. This is an Nmds plot, non-metric multidimensional scaling plot. To see the differences in the composition but by non-metric distances. And here I also show you how I've grouped the samples in these different colors. And from this Nmds plot, you can see that the centroids of the groups are different, and that it seems that this grouping can represent differences in this data set. Our colleagues at the Swedish Defence Research Agency have performed source tracking of the sediments, but I want to perform curated source tracking too. To do that, I looked into literature to find taxonomic groups, mostly genera, but also 2 families. And these are fully associated to municipal sewage and or human gut. And here in this box plot, I display the relative breed abundance of all these groups together as a percentage of the whole data set. For example, in the deep far group, this yellow here, has a mean of 7% sewage taxa, whereas the shallow far group consists of only 1% sewage taxa. Looking at the relative abundance of sewage taxa as a scatter plot instead, to see the individual samples, and on the x-axis here we have instead the distance to A1, which is the effluent pipe location. And Spearman's rank correlation test yielded a significant result for this data, which means that there can be a negative correlation made with significance, which means that the further away from the pipe you get, the lesser amount of sewage taxa you will find. Moving on to differential abundance analysis, for which I performed on taxa level, so not ASV level. Instead, I've grouped all the ASVs together that got the same taxonomic assignment. And by having the E. coli values as the design formula, you can see which taxonomic groups have a similar pattern as the E. coli in the data set. And the taxonomic groups that were yielded significant as an increase in, like an increase in E. coli means an increase of these here are the negative ones, but these here that were positively connected are interesting because they seem to be located at A1 and B3 almost exclusively. There are some extra here, but and from these that are significant, it's also very interesting that, for example, Bacteroides and Prevotella got a very high significance as they are prevalent in human gut. And then we also have Trichococcus and Acinetobacter, which are abundant in wastewater. And then up here, the samples are ordered as due to their Bray-Curtis dissimilarities, which is this tree here. So what we found is that the curated source tracking and differential abundance analysis pretty much show the same thing, which is that there is an effect on the microbial community by sewage. And it also seems that E. coli is a somewhat accurate estimation of the spread. But the big question from the cities remains that, does these E. coli reach the beaches too? So far, we've taken both beach water samples and sediment samples, and it will be a study between March 2019 and August 2021. We're planning to incorporate metagenomics on the sediments to get a broader view of the microorganisms present. Thank you for listening, and also a big thank you to the co-authors of this work, which have worked with this together with me. Thank you so much.
Thank you, Eleanor, for the presentation. Luckily, we could see it at the end. So, is there any question? Either from the... Yeah, David?
Is Eleanor online or Catherine online? Yeah. So my question is, why using directly metagenomics and maybe not amplicon sequencing upfront? It may be a bit more cheap to do amplicon sequencing to get a rapid profile. And so why do you want to directly use metagenomics to have all this data instead, not just amplicon sequencing? We cannot hear you, Eleanor. Have you unmuted your microphone?
Yes, the microphone is on mute, but we don't hear anything.
Okay. Yeah.
Same room with Eleanor, and I'm going to answer the question for her while she has more computer issues, perhaps. If that's okay, Eleanor? Okay. Hi, everyone. This is such an honor to answer questions about Eleanor's work. We are doing Amplicon sequencing, and that is, of course, what we plan to do first on a number of sediment samples collected over these 2 years, as well as also on water samples. And then from those samples, we're going to screen using this. And then if we see interesting correlations from the 16S profiles, then that is when we will proceed with metagenomics. So yes, I agree with you, David. We will take the 16S first.
And thanks for the good reaction per call. We could solve the issue. So there was no way that we could not have the presentation of Eleanor.
No. Thank you so much for your cooperation also. Good teamwork.
We have also a question here by Francesc.
Hello,
yeah.
Or Eleanor, or someone. If what you want is to see the presence of wastewater or gut microbiome bacteria, why don't you compare the microbiome of your samples to the microbiome of the water source instead of looking for the bacteria that you think are common in wastewater, that you see are common in wastewater?
So when we began this project, it was to rule out the sediments as a source for the E. coli. And so we began by taking the sediment samples, which is what was the topic discussed today. And so we could actually see, without needing to match it to the wastewater samples, we could see the background in the sediments against the natural community when we compare it to the other samples where we don't have any wastewater impact. But of course, this would be also something we could add, of course, is to take a sample from the effluent in this wastewater treatment plant. But I think, yeah, we wanted to see a little bit how it was reacting under the natural conditions because we don't know how long it's been there. So it could be a fraction of the wastewater community that is actually able to survive in this slightly saline environment, which is the Öresund.
Thank you. Is there anyone else that wants to add something or to ask something? Well, doesn't seem. Anyway, and with this, we are not gonna have the discussion round now because we went quite over the time today. So I think we all deserve a break now until the next session starts in half an hour. So remember, you can always send messages to the people that presented today if you want to discuss, or also create rooms also in the message section if you want to discuss further any of the topics that they presented today. So see you later in session number 9 at 4:30 and enjoy the break.